Hi dear student, today we begin our lesson with cellulose. Cellulose is the most abundant organic molecule on the planet due to its presence in plant cell wall and slow rate of breakdown. So there are hundreds and thousands of millions of cells in the plant, and all of these cells contain cellulose in them. And number two, slow rate of breakdown in nature, slow decay. Therefore, it is the most abundant organic molecule on the planet. It has a structural role, being a mechanically strong molecule, unlike starch and glycogen. The only difference between cellulose and starch and glycogen is that cellulose is a polymer of beta glucose, not alpha glucose. Successive glucose units are linked at 180 degrees to each other. So in case of starch and glycogen, we studied that both of them are a polymer of alpha glucose. Cellulose is a polymer of beta glucose. Therefore, it is structurally more stable as compared to the starch and glycogen. Now just have a look at the structure of cellulose. Now look at this structure. This structure is said to be beta glucose. Now if the next beta glucose linked with this molecule at 180, this molecule must rotate or flip over to bend to this molecule. Now this is said to be a condensation reaction. Therefore one molecule must be removed, and the bond be linked at oxygen. Now look at here, there is a third. This third molecule must rotate at 180. Therefore what happens in case of cellulose? Cellulose molecule are straight chain. They do not curl up like starch and glycogen. In case of starch and glycogen, just because this is one molecule and this rotate at 180 once again. Now one glycosidic bond is downward, another is upward, another is downward, another is upward, another is downward, another is upward, just because each and every molecule attached to the link rotate at 180 degree. Now look at in case of arrangement of alpha glucose unit in amylose. Now you can see that the molecule does not rotate at 180 degree. Therefore all glycosidic bonds are downward. Therefore they curl up, the curl and coil, turn and coil and they form a spring shaped structure. Now in case of cellulose, each molecule rotate, therefore they are straight chain molecules. Now cell wall is made, or most of the part of cell wall is composed of cellulose. Therefore it's important to study them. A cell wall typically has several layers of fibers running in different directions to increase strength. Cellulose makes up about 20 to 40% of the average cell wall. Other molecules linked to cross-link the cellulose fiber, and some form a glue-like matrix around the fiber, which further increases the strength. Cellulose fiber have a very high tensile strength, almost equal to that of the steel. Dear students, I would like you to remember all these points, because we are going to explain all these points in the next slide. These need to be memorized, or learned. Despite their strength, cellulose fiber are freely permeable, allowing water and solutes to reach or leave the cell surface membrane. Look at the structure of this molecule. Now you can see that these are said to be the cellulose fibers. They are very stronger. The tensile strength of a cellulose can be compared to the tensile strength of the steel. Now they cross-link with one. Now you can see that these molecules are horizontal and these are vertical, make it even more stronger. Now just this point, okay, this one is said to be a cellulose fiber. This is zoomed in to actually understand what happens or what is present inside a cellulose fiber. Now let me tell you one thing more. The diameter of cellulose fiber is around 50 nanometer. Okay, they are made up of many microfibrils, 1, 2, 3, 4, 5, 6, 7. Okay, many, not only 7 but many microfibrils. Now what is inside the microfibril? Okay, just move out of it and you can see that the glycoside, this part, okay, these are actually the cellulose molecule or the cellulose structure. Now you can see that they are straight molecule just because each and every molecule rotate at 180 degrees centigrade. Now if you can see that this is one chain, this is another chain, these chains are actually linked with one another by hydrogen bonding. In our previous lesson we studied that molecule, they have a CH, they have OH, CO or NH groups are said to be polar because there are the partially positive and partially negative charges on them. So these are actually the glucose molecule. They contain hydroxyl group. Therefore, there is a partial negative charge on hydrogen and a partial positive charge on partial negative charge on oxygen and partial positive charge on hydrogen. Therefore, there is a attractive force between these molecules, and a single hydrogen molecule may not have a great effect, but when several thousand or several million these bonds come together, then they form a very great effect on the molecule, making it even more stronger. Okay, now look at this structure. This is the water molecule. Now you can see that the water molecule, hydrogen is partially positive and oxygen is partially negative. Now you can see that there is a bond. A bond can be established between positively charged hydrogen and negatively charged oxygen. This bond is said to be hydrogen bonding. Now this molecule is said to be a polar molecule. Polar because there is a dipole. This is said to be the positive pole, oxygen is said to be the negative pole. Okay, and these actually attract one another. Okay, so now the bond that holds two glucose molecules with one another is said to be the glycosidic bond. The bond that holds one chain with another chain is said to be the hydrogen bond. Okay, now dipoles we just studied in the previous lesson. Okay, for example, look at here. Di means two poles. Okay, water molecule is said to be a polar molecule because it has a dipole. Okay, positive, partial positive or delta positive charge, delta negative charge. Okay, now dipoles and hydrogen bonds. When atoms and molecules are held together by covalent bond, they share electron with one another. Each shared pair of electrons form one covalent bond. For example, in a water molecule, as we observed above, two hydrogen atoms each share a pair of electrons with an oxygen atom, forming a molecule with the formula H2O. In water, the negatively charged oxygen of one molecule is attracted to positively charged hydrogen of another, and this attraction is called hydrogen bond. It is much weaker than covalent bond but still has a very significant effect. Look at this structure. This is hydrogen molecule. You can see it is partially positive. This is oxygen molecule. You can see partially negative charge. Okay, now what happens just because it has two, the molecule is said to be polar. Okay, and it can really dissolve polar molecule solutes in them. Okay, now this positively charged can be attractive to negatively charged oxygen of another water molecule. Okay, the bond that holds two molecules of water with one another is said to be a hydrogen bonding. Now lipids are esters. Okay, esters are molecules, or esters are the molecule formed as a result of acid and alcohol reaction, or when acid react with alcohol, esters are formed, and lipids are actually esters. Lipids are the combination of acid and alcohol. For example, fats are said to be the lipids. Fatty acid and glycerol combine together to form fats. Okay, so fatty acid are acids and glycerol are alcohol. They are all organic molecules which are insoluble in water. The most familiar lipid are fats and oils. Fats are solid at room temperature and oils are liquid at room temperature. Fatty acids. Now, fatty acids are a series of acids, some of which are found in fats. They contain acidic group COOH or carboxyl group. Okay. The larger molecule in the series have long hydrocarbon tail attached to the acid head of the molecule. As the name suggests, the hydrogen bond tails consist of a chain of carbon atom combined with a hydrogen. The chain is often 15 or 17 carbon long. Look at the structure. Now you can see that this part is said to be hydroxyl group. Okay, because it contains carbon, oxygen, and hydrogen. This part is said to be hydrophilic. It means that this part can dissolve in water. Now look at down here. Okay, all these molecules are said to be hydrophobic hydrocarbon tail, because it cannot be dissolved in water. Now you can see that all the bonding in this molecule is said to be single covalent. Okay, all the bonds between carbon and carbon are single covalent. Therefore, this molecule is said to be saturated fatty acids. Okay, now look at here. You can see that the carboxyl group is present right here, and it is also present right here. Okay, now you can see that 1, 2, 3, 4, 5, 6, 7, these bonds are single covalent bond. Okay, now you can see there is a double bond. Now, if there is a double bond, it means that it is unsaturated. Okay, a molecule is said to be unsaturated if it contains one or more than one double bonds in them. Now you can see that there is 1, 2, and 3 double bonds between carbon and carbon. So this molecule is said to be unsaturated fatty acids. Now you can see that there are more than one double bonds. Therefore, this is said to be polyunsaturated. Okay, if it contains one double bond, it would be called monounsaturated. If it contains more than one, it would be said to polyunsaturated. This molecule is said to be polyunsaturated molecule. Now, saturated and unsaturated, we described them in the previous slide. Saturated molecule are said to be so said because there is a single covalent bond between carbon and carbon. Okay, now unsaturated is one that is at least one double or triple bond between carbon and carbon. The tail of some of fatty acids have double bond between neighboring carbon atoms like this, carbon-carbon double bond. Such fatty acids are unsaturated because they do not contain the maximum possible amount of hydrogen. Now it's very important. They do not contain the maximum possible amount of hydrogen. They are unsaturated lipids. Let me tell you one thing over here. Unsaturated fats are much useful as compared to saturated. Animal fats are said to be saturated, and it leads to heart diseases and coronary heart diseases, okay, and high blood pressure, etc. Unsaturated fat readily dissolve in the body and less likely develop heart diseases. Double bond make fatty acid and lipids melt more easily. For example, most oil are unsaturated. If there are more than one double bond, the fatty acid or lipids are described as polyunsaturated. If there is only one, it is monounsaturated. Animal fats are often unsaturated and plant lipids are often unsaturated. Alcohol and ester. Let me define the term alcohol. Alcohol or the molecule formed when hydrogen in alkane family is replaced by OH group. For example, the first member of alkane family is methane, CH4. Remove one hydrogen, now it becomes CH3. Add OH, it becomes CH3OH, methanol. Alcohol are formed when hydrogen in the alkane family is replaced by hydroxyl group. Okay. Now, what are ester? Ester, we explained that esters are the molecule formed as a result of acid and alcohol reaction. Alcohol react with acid to form esters. Alcohol are a series of organic molecules which contain hydroxyl group OH attached to a carbon atom. Glycerol is an alcohol with 3 hydroxyl groups. The reaction between an acid and alcohol produces a chemical known as ester. The chemical link between acid and the alcohol is known as ester bond. Ester bond is actually the bond that holds an acid to alcohol molecule, or ester linkage. Triglyceride. Okay, if you look at the structure of this molecule is said to be, for example, this molecule is said to be glycerol. It contains 1, 2 and 3 hydroxyl group. This whole molecule is said to be glycerol. Okay, it contains 3 hydroxyl group. Now you can see that this is said to be the fatty acid. One fatty acid, another fatty acid, another fatty acid. Now this is alcohol and these are fatty acids. What happens? This should be removed. One water molecule, another water molecule, another water molecule. So 3 molecules must be removed. And what would happen? The hydrogen left will link with the acid. Now look at here. This is actually the oxygen atom that is holding the head of the molecule to the tail of the molecule. This bond is said to be ester linkage or ester bond. 1, 2, 3 ester linkages or ester bond. For example, this is the skeletal structure or skeletal molecule or the main or the backbone, and these are the fatty acid link. This red line represents ester bond or ester linkage. Triglycerides. The most common lipids are triglycerides. These are fats and oil. A triglyceride is an ester formed by a fatty acid combining with the alcohol glycerol. Glycerol have 3 hydroxyl groups. Each one is able to undergo a condensation reaction with a fatty acids. When triglycerides is made, the final molecule contains 3 fatty acid tail and 3 ester bonds. The tail can vary in length depending on the fatty acid used. Okay. Now you can see that this point represents CH2. CH2, 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 CH2 and so on. Okay. The length may contain 15 or 17 carbons in this whole chain. Okay, now look at here. This is said to be glycerol, this is fatty acid, and this is ester bond. For example, this is actually alkane molecule C3H8. Now when a hydrogen in the alkane family is replaced by hydroxyl group, one hydroxyl group, another hydroxyl group, another 3 of the hydrogen in alkane family are replaced by hydroxyl group. Therefore, this is said to be glycerol, or you can say propane triol. Okay, it combined with the fatty acid and triglyceride molecule are produced. This whole molecule is said to be triglycerides. Now, what is the role of triglycerides? Lipids make excellent energy reservoir. They produce more energy than it is produced by carbohydrates or proteins. A given amount of lipid will yield more energy on oxidation than the same mass of carbohydrates. Fat is stored in a number of places below the dermis, skin, and around the kidneys. An unusual role of lipid is as metabolic source of water. For example, the desert kangaroo rat never drink water and survives on metabolic water from its fat intake. This is one of the animal, desert kangaroo rat. They never drink water, just there are certain chemical reaction inside the body that produces water, okay, from the fat intake. Phospholipids. Okay, this is actually the special type of lipids. Phospholipid is a special type of lipid. Each molecule has an unusual property of having one end which is soluble in water, the phosphate group. And so it is actually a phospho means phosphate, lipids means fat. Phosphate are hydrophilic, they love water, they can dissolve in water, and lipids hydrophobic, they hate water. This is because one of the three fatty acid molecule is replaced by phosphate group. This is polar and can therefore dissolve in water. The phosphate group is hydrophilic, water loving. The two remaining tail are hydrophobic, water hating. Hydrophilic head lie in the water solution on the outside of the membrane, while the hydrophobic tail form a layer which is impermeable to hydrophilic substances. Look at this structure. Now this is actually the glycerol. Glycerol we studied in the previous lesson. For example, right here we studied that this is the glycerol and these are the fatty acids. Now what happens if we replace this one fatty acid by phospholipid? The molecule is said to be phospholipid. Okay, this is the fatty acid. If we replace this fatty acid with the phosphate group, now this whole molecule is said to be phospholipid. Instead of triglycerides, now we call it phospholipids. Cell membrane is actually made up of this molecule. Now the tail is composed of phosphate group. Phosphate is hydrophilic. The head is actually composed of phosphate, and phosphate are hydrophilic, water loving, dissolves in water. Now look at here, this is the tail, okay, and there might be a double bond over here, therefore this is said to be hydrophobic tail, water heading, okay, and this whole molecule is said to be phospholipid, and cell membrane are made up of these molecules. Now proteins are actually the molecule made up of amino acids, and there are four different types. On the basis of structure, there are four different types of proteins: primary structure, secondary structure, tertiary structure, and quaternary structure.